Kevin, and thanks, Emma, and lovely to see so many familiar faces and see people who came to the first conference that we held in Newcastle in 2011. It seems a long time ago in a way, but in another way it seems a very short time in view of what's happened in that period of time in our understanding of a disease and the treatment of disease. So what I've been asked to talk about today is a phenomenon called penetrance, and I'll try to explain to you, as I hopefully go slowly, through genetics. So some of you will know that I've been interested in this disease for over 30 years now, and I first became interested because I met one individual who suffered from a disease called Sean McCowie, and Sean is a patient who was on dialysis in Newcastle, and none of what I say is confidential. Sean's given me permission to say anything I want about it, well, not absolutely anything I want about him. So Sean is part of this family in Newcastle upon Tyne, which has suffered severely from this disease. And this is the pedigree for this family. And some of you may not be familiar with family trees, so let me run through you what a family tree is. So the squares are males and the circles are females. If a symbol here is filled in, it means that the person has been affected by the disease. If there is a line across the symbol, it means that person is deceased, has passed away. If there is a symbol like this, it means that these individuals are identical twins. So that sign there means that they're identical, that means that they're twins. A sign with a symbol with a little circle inside it means that somebody has to carry the genetic abnormality in this family, but has not yet developed the disease themselves. So in Sean's family, and here is Sean, who is the only surviving affected individual in this family where 8 members have been affected by the disease, you can see that in this family there are 2 individuals, one still alive and one who's passed away, who must carry the genetic abnormality because they have passed it on and inherited it and passed it on. And therefore they must carry the same genetic abnormality that their parent and their child have, but they haven't developed the disease themselves. We call this phenomenon non-penetrance, i.e. the disease has not manifested themselves in that individual despite them carrying a genetic abnormality. Now we see this in other families, and I'm going to show you a much larger family, which some of you here will be familiar with, where this is another seen in that family as well. There are people who carry the genetic abnormality, but throughout their lives have not developed the disease. There are other things in this family that are very common to other families with the disease, in that everybody who got the disease, despite the treatment they had initially when they first got the disease, they did not recover renal function. They went into kidney failure. 3 people in this family also got a kidney transplant, and the kidney transplant in all those 3 was unsuccessful because the disease came back immediately in the kidney transplant as a result of the underlying disease. And Sean, up to recently, had thought that he would end his days on dialysis because he thought there was no prospect whatsoever that he would ever get a transplant, that he would die on dialysis. But I'm delighted to tell you that Sean, I don't think Sean's here, but Sean 18 months ago had a transplant with eculizumab and feels better than he's ever done for years and years. So he's one of the many individuals in the UK who has been successfully transplanted with eculizumab who thought they would never, ever get a transplant. So I've introduced you to the concept of what non-penetrance is. So this is a phenomenon whereby somebody carries a genetic abnormality that is present in other people of their family, but they do not develop the disease that the other people in that family develop. So the question is, why does this occur? Why don't they get the disease? So the hypothesis, the reason that we have put forward is that for an individual to develop the disease, atypical aHUS, you need at least 3 factors. So you will need a predisposing significant variant. We would often call that a mutation. I'm going to tell you what I mean by a mutation in a little while. So you need an underlying rare genetic variant, which we would have called previously mutation. You will need on top of that another adverse modifying variant in another gene, maybe in the same gene, that is going to increase your risk of getting the disease. And then on top of that, you're going to need a trigger. So let's say that somebody carries one of these predisposing variants, but doesn't have an adverse modifying variant, or never comes face to face with a trigger that would precipitate the disease in their life. You can start to see why somebody might go throughout their life, despite carrying the genetic abnormality, and not develop the disease. Okay, so store that information for a little while. To understand this from a point of view of modifying variants, I'm going to have to give you a little bit of information about genetics. So, we all have 22 pairs of chromosomes, and an X and an X chromosome if we're female, and an X and a Y chromosome if we're male. We inherit all our genetic material from our mum and our dad. And for every gene, we inherit one copy from our mum and one copy from our dad. So every gene we have 2 copies of every gene. So Kevin's been talking about factor H, factor I, CD46. There is a gene for every one of those proteins, and we have 2 copies of that gene. The chromosomes, which contain the nuclear material, DNA, which goes to make up genes, lie within the cells in a central portion of a cell called the nucleus, right in the middle. So if I take out the nucleus and I blow up the chromosome, and then I start to blow up the DNA, you can see this helical structure that Crick and Watson described famously years ago of DNA. DNA is, in essence, a chemical code. So here is that helical structure that Crick and Watson described many years ago. And in essence, it's a very simple chemical code consisting of 3 letters, A, T, C, and G. And each of these 3 is a different chemical. We call it a nucleotide, but you don't need to know. Just remember that each of these is a different letter. Okay? So let's put these letters together in a code. The way the code works is that each 3 pairs of letter forms an individual code. It codes for an amino acid. So here we have a gene, and here we start to see the code. That codes for one amino acid, that codes for another, that codes for another. Let me take that a little bit further. So each set of 3 letters is a code. And it's a code which goes to give the instructions for a building block called an amino acid. And it's like somebody saying to a bricklayer, red brick, white brick, yellow brick, and put them on. And the code runs along, so it's like reading the code. The code will tell you which block to take. So there are 20 amino acids, and I've likened the amino acids here to Lego bricks. So each color is a different amino acid. And those of you who are astute and who are good at computation in their mind, like Len, will have worked out that with 4 different codes, there were 64 possible permutations of 4 letters. Is that correct? Yes, David. Good. There aren't 64 amino acids, and I'll tell you why there are not 64 amino acids in a minute. So when the amino acids are put together, and I've taken an analogy here of James May's Lego house, you get a protein. So here's James May's Lego house protein. So our bodies are in essence made of protein. We are walking protein. And protein can be something I can feel, like I touch my skin. My skin is made up of protein. But proteins can also be in solution. So when we get out milk every morning for our breakfast, if we have cereal, and we pour it in, those are proteins in solution. There's protein and several thousands of proteins. There are at least 2 million different proteins in the human body. So there must be at least 2 million different genes that's stored on those chromosomes. There are 6 billion different codes that are made up, which you have to make up. It's a huge number of codes. Keep that in mind. Okay, so this is the reason there are not 64 amino acids. So what I've given you here is the three-letter codes, TTT, TTC. And here, each one of these is a symbol for one of those building blocks called an amino acid. So this is shorthand for phenylalanine, shorthand for leucine. One of the things you'll start to see here is that the same building block can have more than one three-letter code coding for it. So the bricklayer could get CTT and he'll throw in a leucine. He might get CTA and he'll throw in a leucine. He might get CTC, he'll throw in a leucine. So several instructions can code for the same building block, all right? The other thing to notice is that if I just change one letter in this three-letter code, it will result in a new building block instruction being given to the bricklayer. So if here you can see the four three-letter, well in fact six, these are the four three-letter codes that code for leucine. If I just change the T in this to a C, it will start to code for proline. So just one letter change will result in a different brick being put into James May's house. So what might that look like? Well, let's stick with leucine and proline. Here is the molecular structure of leucine. Here are carbon atoms and hydrogen atoms and nitrogen atoms in leucine. So we change one letter in that code, and the brick that is put in, which I've said here is a free brick looking like that, will change to this. You can see that the structure of that brick looking here, I've got a great laser pointer. Kevin, you've got a laser pointer. They look completely different. So if I put this brick in James May's house and replaced it with that brick, you can see that the actual brick would look different. Thanks, Kevin. That's great. All right, so you can start to see how if I change just one letter in that code, it will result in a different brick being put in. I want to come back, however, to this code to show you some other codes. There are two other codes that are really, really important. In every protein, in every house that is built by the bricklayer, the first brick that is laid is always methionine. And there is only one code for methionine, which is ATG. So the first brick in a house always has to be this. So if we get a one-letter change at the beginning of the code for that house will never get built, because the bricklayer will never get the code to put that one brick in to start off with. There is also a code for the bricklayer to stop laying bricks, and that code is TGA. Now, let's say that we have a code elsewhere, and I'm trying to see one. All right, let's say we are planning to put in a leucine, all right, TTA. And somehow the code changes from TTA, where leucine is meant to be put in, to a TGA. The bricklayer will down tools and will stop building the house. So there's an error there. That protein, which was meant to be built, will just stop being built. It will become redundant. The house will never be built. And this is a sort of introduction to you for genetic variants. Okay. So what is a genetic variant? So here I've got my series of three-letter codes, each one coding for this different building block amino acid, which is going to make the house. So if you can see here, this is all identical. This is the gene. I've changed the C there to a G there, and that will result in a different brick being put in. So the change to this code leads to a different brick, a different amino acid. What might the result of this be if I put a different brick in that protein? Well, it might actually have no effect whatsoever, because the brick that I've replaced one brick with another brick is so similar that it's not going to look, but it might be a little bit of a different color, but it's not going to change anything in the protein. So the function of that protein will not change. It might just change it a little bit, but not enough that it's going to cause major abnormalities to the individual, and I call this a modification. So brick changes what the protein does, but only to a very minor extent. And this could either be an improvement in what that protein is doing, or it could be a deterioration, but it's only a minor change. And this is quite common, and it's usually found in more than 1 in 50 individuals we will find variants. These variants explain why we all look different, because if our genetic code was identical, we would all look the same. We would all be identical multiplets. But our genetic is different. We have different variations, and that's why we're different. If the change in the brick is a big change, what I call a major change, with a big deterioration, usually it's a deterioration. It's usually not a big improvement. If it's a big deterioration, what the protein does, then it can have a really harmful effect. And that's that part of that triplet where I said a significant change. That's what we're looking for. And usually they're much rarer than the common variants found in less than 150 individuals. And up till recently, it was known as a mutation. So these were the changes that we would call, oh, that individual's got a mutation. Coming back to what I told you earlier on, we have one gene from our mum, one gene from our dad, and we have 2 copies for every gene. When we get a variant in only 1 copy of a gene, we call that heterozygous. And in most of the genetic abnormalities that cause aHUS, it is a heterozygous change. It's 1 abnormality out of the 2 genes. Rarely you can get the same fault in both genes, and that often happens when your parents are related, because they carry very similar genetic material. And we call that a homozygous change, much rarer seen in atypical aHUS. Okay, so I already explained to you that we all differ because there are variants in our genes. So let's take my gene here and add in a couple more variants. So I put an A there that differs from a T, kept the C and the G, and the C and the T. I put in a C and a T. So because of what we are, let's call this gene Senate House. And there's certain nomenclature which we keep strictly to, and I go on and on about the nomenclature to people like David. And the nomenclature is that whenever we call a gene, we put it in italics. So if you look at them in papers, we have to be very precise. And I'm a bit of a pedantic about this. David will vouch for that. I am very pedantic about this, but it's very important. And those 2 differences in the same gene we call haplotypes. I'm going to come back to that later on. So the 2 haplotypes of this gene, the nomenclature we give to this is Senate House A, C, G, written under there, and Senate House T, G, T. Now, in most populations, there are more than 1 haplotype, so we might get variant. Let's, for simplicity, say that these are the 2 haplotypes that are found in the Senate House gene in the population. And we will find when we look at the population of a whole and we start to do the reading of this, that we will find proportions of a population. So we might find 60% of the population have that form and 40% have that form. And so we can start to give percentage values for populations of having those 2 forms. Okay. So how do these variants actually come about? Let me go back to this. Why does this change to this? Why do we get a variant? Well, radiation, ultraviolet, chemicals can cause our DNA to change spontaneously. By far the commonest thing is when our cells divide. So every time our cells divide, the 6 billion letters that is in the genetic material has to be copied. And you can think, if you have to copy 6 billion letters, there's a chance you're going to make a mistake in copying 6 billion letters every time. Now, the body has very sophisticated proofreading mechanisms. So every time our cells divide and copies our DNA code, there's a proofreader who goes along to check that there's no errors in copying this. It's a chemical proofreading system, but sometimes that proofreading system will miss things. So here's a cell dividing, perhaps, and what happens is that comes through. Now, the cells in our bodies are constantly dividing. Our blood cells are constantly dividing. It doesn't really matter, except in one crucial situation, if we develop a variant in other parts of our body. So I suspect that in parts of my muscle, parts of my brain, there are lots of variants being produced, but they're not being transmitted to other generations because they're not in sperm cells or ova. But in sperm cells or ova, if a variant develops, then there is a chance that it will then be passed on to the next generation in sperm. Unfortunately, gentlemen, we are responsible for most mutations because sperm divides much more rapidly than ova. And so because sperm develops with sperm cells, there is much greater chance of variants developing in sperm cells than ova. So that's the reason. Number one situation where in our other cells that a variant could be an abnormal is in genes that stop cancers being developing in certain cells. So the majority of cancers develop because of a variant developing in a cell type in a gene that is responsible for stopping cancers developing. Okay, now Kevin talked to you earlier on about the brakes on the complement system. And two of the major brakes are called factor H, this protein that is in solution, but binds to cells and prevents complement attacking our own cells, and this other regulator brake called CD46 that is within cells, that's resident there, that is available immediately, but complement attacks the cells to stop the complement. So the gene for factor H is in italics CFH, and the gene for CD46 is in italics CD46. Okay. So we know from studies in the normal population and in individuals who've got atypical aHUS that CFH has 8 different haplotypes in the population. We know from studying people with atypical aHUS and comparing those with people who don't have aHUS, what we would call normal controls, that there is one haplotype that protects against atypical aHUS and another haplotype that seems to increase the risk of aHUS if you also carry a rare variant, a mutation, either in factor H or one of the other genes. So CFH, C80AAG, which is also called H2, protects against aHUS. And we know the reason it's found protects is because it's found in 35% of aHUS versus 57% of normal controls. The other haplotype, which is CFH-TGT-GGT, H3, increases the risk, and we know that it increases risk because there's a significant statistical difference between the percentage of patients with aHUS who carry that and individuals who don't, the normal controls. Within the population, the CD46 gene, there are 4 different haplotypes, and we know there's one particular haplotype called CD46GGAAC, which increases the risk of atypical aHUS. And the reason we know that is when we look at aHUS, 44% of people have it versus normal controls only having 21%. Now, but that information has come from taking a whole series of different patients with atypical H and comparing them with a normal population. So, let me show you a case history to try and exemplify what I've been talking about in terms of significant variant, adverse modifying variant, trigger, and disease. So, this is an individual who I know well, who's given me her permission to disclose, I'm not going to mention any names, this information, discuss it with you today. So this is a female who at the age of 22 developed a rare condition whereby you get clotting of the main vein of the liver. And she was admitted to a liver unit at King's College Hospital in London, and they thoroughly investigated this individual. And people who get this condition, you can sometimes find an underlying abnormality that explains why they've got this clotting. It stops the liver functioning. And when it stops the liver functioning in a very severe way, the only treatment is to have a liver transplant. So this individual underwent a liver transplant, which was very successful, no complications, and as with kidney transplants, we have to use immunosuppression, and she was put on a drug called tacrolimus and maintained on it without any problems whatsoever. The level of the blood, of the drug level of tacrolimus was fine. So, 3 years later, she becomes pregnant. The pregnancy is successful, but 3 weeks after her delivery, she develops atypical aHUS. She's treated with plasma exchange without any recovery of renal function and has to start dialysis. We were sent samples of her blood and her DNA to analyze to see if we could see why had she developed atypical aHUS. And when we measured levels of those substances that Kevin already mentioned, like C3, factor H, we could find no abnormality whatsoever of those. We looked at all the genes that are known to be associated with atypical aHUS, and we couldn't find any abnormality of those either. And we looked for autoantibodies that Kevin has described against factor H, and we couldn't find any abnormalities of those. So we thought, well, what about the donor, the individual who unfortunately had passed away, who had consented to have, or family had consented for that liver to be used for transplantation? It's possible to get information about donors when it is relevant to an individual who's received transplant medical history. So we were able to find out that the donor had died of a brain hemorrhage, and in this donor, the kidney function was completely normal. There was no evidence that this individual had any kidney abnormality, and there was no family history either. We were able to get DNA, which is stored routinely from all donors in the UK for this very reason, for what we're doing here. And we screened the donor DNA, and we found that in the donor had a rare variant, a mutation, in one copy of the 2 factor H genes, and this variant was leading to the bricklayer stopping laying bricks from that copy of a gene. So this individual, until the time of death, had half normal levels of factor H, came to no harm whatsoever herself. So, in the individual who received that liver, who then went on to develop atypical aHUS after pregnancy, could we find any adverse modifying variants? And we screened both factor H and CD46, and we found that both copies of this individual's CD46 gene had the at-risk CD46 haplotype. Okay, so let's put that all together. So we here have an individual who carries an at-risk, and we call this homozygous, CD46 haplotype, which would increase your risk of developing atypical aHUS if you had a significant variant and mutation. Up to the age of 22, this person didn't have a significant variant, but then underwent a liver transplant and acquired, through the liver of the donor, the significant variant in factor H, resulting in half normal production of factor H. For another 3 years, despite carrying these 2 predisposing factors, the individual was fine until pregnancy, and pregnancy, Edwin's going to be talking about pregnancy as a risk factor, until pregnancy ensued and the disease manifested itself. So I hope that's a way of seeing how this works out for individual patients. A nice story about this is that this individual, obviously, as I said, went on to dialysis and then went on to get a transplant from her mum with eculizumab, and has had the transplant now for a year and is doing brilliantly with brilliant transplant function. So a nice end to that story. So Kevin's already shown you this slide earlier on. From what I've been saying, you will understand that studying families has been pivotal in what we have learnt about this disease. Families have unlocked the disease. 30 years ago, when I first started looking at the disease, we didn't have a clue what caused it. There were all sorts of hypotheses, and it was families, in particular a family that I will mention in a minute, that unlocked the key to understanding what causes. And the first mutation that we ever found was in a family with factor H. And now we've gone on to screen 56 families in total. So we've got 56 families where more than one individual has been affected by the disease, which we've been studying over the last 30 years. And this is bringing it right up to date to 2015. And it gives you the percent of abnormalities that we've found in families. We've used the most up-to-date methods of screening genes. So we were using what I would call cutting-edge technology to try and identify in these families. Despite using the best techniques, there are still 30% of our families where we do not know what the underlying abnormality is. We have found several families where we think there's a novel gene. There's something completely new. In one of them, it is a complement gene. In two, it looks as if it may be a non-complement gene. And that's of relevance because Kevin mentioned this other gene called DGK epsilon, which is a gene that is no way part of a complement pathway. And we now know that most individuals who we find an abnormality in this gene, DGK epsilon, because it is not a complement gene, will not respond to eculizumab. So it's very important that when somebody is started on eculizumab, that we screen for this gene to be certain whether or not they may have this abnormality and therefore would not respond to the drug. So what I've done here is taken the whole of a cohort that we've been studying over the last 30 years, and I've compared the number of families who have what I call a rare variant, a mutation in the genes, and the number of individuals without a family history, who do not have a member of a family who've been affected, who've got a variant in that same gene. So for factor VIII, there are 15 families, 57 individuals. 21%, therefore, that is 21% of the total. CD46, 10, 23. C3, 5, 22, 19%. It's interesting that with relatively small numbers, these are all, I would say, in the same ballpark. A take-home message is that there are going to be, in these people who have a variant, the likelihood that they are the first person in that family to ever develop that variant is very, very unlikely. And it's coming true as we start to screen more and more people that we see numerous families where we find one person and we screen other people in this family and we find more and more individuals in that family who carry that genetic variant. And I want you to remember that because I'm going to talk about this phenomenon penetrance in a minute. Two genes, factor I and factor B, we find very low levels of familial. So we've got only two families. We haven't got any families in Newcastle, no numbers. DGK epsilon is very different. High level of genetic familial abnormalities in this. And this is the only one of the diseases that is fully penetrant. By fully penetrant, I mean if you have two abnormalities in both copies of your DGK epsilon gene, you will get the disease. We have not ever found anybody with DGK epsilon who's got abnormality who has not developed the disease. It's very rare, and it's homozygous. Okay, so this is one of my favorite families, isn't it, Margaret? So this is a family that comes from the southwest and is probably well known to many of you in the audience. It is undoubtedly the biggest family in the world sadly affected by this disease, and it was first described in a paper by Edelstein and Tuck, who described this paper in 1978. And 30 years ago, my colleague Paul Warwicker here was working on another genetic disease called the psychoureteric reflux. And this was his PhD project. And one day he came to see us and he said, this psychoureteric project is not going very well. And he said, isn't there anything else I could do apart from this project? And I said to him, well, look, there's this family in Newcastle who've got this other disease called atypical aHUS. What about having a look at this family as well? So Paul said, great, I'll go away and I'll have a look. And he disappeared, and then a few weeks later he came back and he said, I found this other family down in Exeter. And so Paul tells us to, Margaret, tell me whether it's true. So Paul went down to Exeter to see this family, which was the original branch of a family, and Paul said, do you know what, they told me when I went to see them that was another family living nearby who they thought had the same condition. So Paul, when he was down in Exeter, found this family. And initially it wasn't possible to establish a common lineage for this family, but Paul, by going through and with help from the members of that family, was able to establish a common lineage for that family. And that was a family that Paul used as one of 3 families to do the first genetic study in atypical aHUS. And it was the first ever factor H mutation that we found. So here we have the mutation. Factor H with a code change of C to G in that position, resulting in a change in an arginine to a glycine in position 1215 of one copy of the factor H. And that was the first ever mutation ever described. More recently, in 2012, you can see that the family has grown substantially, and this is an enormous family. The family, in fact, is, we think, much bigger than this, and we think there are branches in Swansea and Manchester, and we think there are other branches of the family elsewhere. But in 2012, we started to look at this family again, because as in Sean's family, there are individuals in this family who we know, because we screen them, carry the genetic abnormality, but throughout their lives have not developed the disease. So in 2012, there were 25 individuals in this family who had been affected by the disease, and screening in a church hall somewhere in the southwest of England, there was a big party, and all the people came together and blood was taken, and they sent it up to us and we screened. We found there were 18 unaffected carriers at that time. What's interesting is when you look at the 25 individuals who were affected by the disease and plot out the age, so this is the age at which they first developed the disease. There's a cluster of individuals who developed the disease very early in childhood. Each line represents a single individual getting the disease. Another cluster of individuals who developed it in late teens and early adults. And then another larger, more prolonged cluster of individuals who first developed disease in the 50s, and the eldest person in this family to develop a disease for the first time developed it at the age of 79. So using this information, we were able, using specialist genetic techniques, to calculate what was the risk of developing the disease in this family at certain ages. So we worked out at the age of 4 for 10% risk, 27, 29%, 60, 51%, and 70, 64%. The difficulty in this family with the eldest person developing it at 79, it's very difficult to say to a 60, 70-year-old that you can't really say there's no risk for you whatsoever. All we can do is give the figures that we have here. But for an individual who carries the genetic abnormality, there is probably at best in this family, as far as we can say, a 64% risk of developing the disease during the lifetime. Okay, so what we did, we were able to get DNA from 8 of those 25 affected individuals. So here are results for the affected individuals, and here are results for the unaffected individuals at that time. Let's look first at CD46. And I told you that there is a CD46 haplotype that increases in other patients, not specifically in this family. We're going to look and see in this family whether it holds true. But there is a CD46 haplotype that increases the risk of you getting a disease if you carry a mutation in another gene. So we know that this family, all the people here have got this variant in factor H. When we look at that, remember there's 2 copies of every gene, so we're going to have 2 haplotypes. when I've shaded it out, shading shows where the at-risk haplotypes, so there's at-risk, there's the at-risk, there's the at-risk. You can see just looking at this, there is no difference in the frequency of this at-risk GGAAC haplotype in the affected individuals compared to the unaffected individuals. So in this individual family, there seems to be no evidence whatsoever that that haplotype is changing the risk of developing the disease if you've got the factor H variant. Now, 2 copies of factor H gene. Because everybody here has got that genetic abnormality, one haplotype has to be identical because they've inherited the same haplotype. And as you can see, everybody, affected and unaffected, has got this TGT-AAG haplotype. And that TGT-AAG haplotype is the haplotype upon which sits the factor H mutation. It's in that gene. But what's of interest is what's happening on the other gene. So, I have shaded here for you the at-risk haplotype that increases your risk of developing the disease if you've got the mutation. 4 in the people who affected it, 1 in the people who didn't get the disease. What was even more powerful from a statistical point of view when we looked at this was to look at the protective. So here, in red and underlined, is the protective haplotype. And the protective haplotype was found in 6 of the individuals here, but nobody who got the disease had got the protective haplotype. So what we were able to take away from this study was that in this family, the one thing that we have found that seemed to be determining whether or not you manifested the disease is what the haplotype was on the other copy of the factor H gene, the gene that didn't have the mutation in it. Now, why might that be functioning? One of the things that we are beginning to realize is that different haplotypes of genes, particularly factor H, are associated with different levels of production. So it's almost like one bricklayer is laying more bricks faster and is building more of a factor H. And so there are studies coming to them that suggest that this haplotype actually results in more production and therefore marginally higher levels of that protective protein, factor H. So you've got a little bit more protective protein to protect against your mutant form of factor H. Okay, my last slide. Can we use the information that we've taken from this single family for all aHUS families and all individuals and their relatives? So, those of you who I've met and those of you whose families I have met and talked to, who we know are unaffected carriers, what I say is that using the information from this family, that we quote a 64% penetrance at age 70 as a worst case risk for unaffected carriers. But I think the family I've just shown you is unique. I think in most families, for the reasons I've told you already, that there are many individuals who we know carry a rare variant, but whom we screen multiple other people as a family and find no one, that the risk is substantially less. In fact, what I'm trying to say is that families like Sean's and the family from Exeter are unique, and what we should be asking there is, why is the disease so penetrant in that family when it doesn't seem to be as penetrant elsewhere? So what I use this figure is a worst case risk, and I say to most people, for most people, I think the risk for your unaffected carriers in your families is going to be substantially less than that. The information we've got from this family, however, does urge me to be cautious in using CD46 and CFH haplotypes in other families to predict what's going on, because as I showed you, in this family, the CD46 haplotype in no way predicts whether an individual was at risk of developing the disease. So there has been some enthusiasm for perhaps haplotyping unaffected carriers and saying you've got a so-and-so risk. And what I always say is never say never. So I'm very cautious about saying to an individual, we'll do your factor H haplotype and your CD46 haplotype and we'll work out an at-risk and you'll never develop a disease, because that's just asking for somebody to go and develop the disease and to ask me why I told them that. So I think that perhaps what is most important in families where we know that one individual carries a genetic abnormality is awareness that other people are at risk, and awareness of that risk is paramount with early action in the UK, because we now have a very effective treatment for the disease. And so the Southwest family, since 2012, when I did that, 2 new people have been affected by the disease. Both were diagnosed within 24 hours, I would suggest, of getting the disease. Both received eculizumab within 48 hours. Both have normal kidney function now. And both those individuals, I suspect, would have been at high risk of going into end-stage renal failure and being on dialysis. So being aware of a risk and using, and great to see that the cards are in the packs, using the cards that aHUS UK have developed that you can carry and give to healthcare professionals to say, look, I'm at risk, these are the tests you should do, and if they're normal, I want you to send me to a kidney unit, is really a paramount, because the one thing that is becoming obvious as we're starting to gain experience from running a national service is that the sooner you're treated with eculizumab, the better your chances of recovering kidney function. So, thank you very much for listening to me. I hope I have not caused widespread confusion. Thank you.